Thank you for joining us today. I'm Eddie Stevens, CSO at Metrion Biosciences, and I'll be chairing today's webinar on trigeminal neuralgia. I'm delighted to welcome Professor Michael Gold, a professor of neurobiology at the University of Pittsburgh, and Metrion's Director of Neuroscience, Dr. Tony Rush. They'll present data on sensory neuron ion channels, sensory neuron excitability, and mechanisms underlying trigeminal neuralgia. If you have any questions for the presenters, please enter it into the questions panel and we'll answer as many questions as time allows after both presentations. So if you're not aware of Metrion, we're an ion channel CRO based in Cambridge, UK, and we have a range of assays, so ion channel screening assays from manual patch clamp recordings up to high throughput screening assays using automated electrophysiology and Flipper, cardiac safety screening, including GLP-HERG, and neuroscience assays using electrophysiological recordings of different neuronal preparations. Now I'll introduce Michael. So Michael is a professor of neurobiology at the University of Pittsburgh. He received his BA from Berkeley, PhD from UCLA, and was postdoctoral fellow with John Levine at UCSF. The focus of Michael's research is the neurobiology of pain. He's made important contributions to injury-induced plasticity in nociceptive afferents and their contributions to persistent pain. He's employed a range of approaches to understanding pain pathophysiology, from the study of isolated cells to development of novel behavioral assays, and more recently, the study of clinical populations suffering from persistent pain. So Michael, over to you.
Well, thank you for the introduction, and hello, everybody. And thank you for the invitation to participate in the session today. For those of you that are new to trigeminal neuralgia, it's not certainly in terms of prevalence relative to low back pain or NEOA, for example, it doesn't match that kind of prevalence, but in terms of severity, it is still known as one of the most severe pain syndromes, and is still referred to as a suicide pain syndrome because, in fact, a number of people do still unfortunately choose to take their lives because the pain is so intense. And so because of that, I think it is still a critically important pain syndrome to study. In terms of the disclosures, I have support, at least for a little bit longer. We'll see how things go, from the NIH, DoD, and the Block Foundation. And I'm currently the treasurer for the International Association for the Study of Pain, but nothing that I'll be talking about today will have any impact on, that the source of funding will have any impact on. Just as a general background for my approach, I consider what we do in the lab as taking sort of a reverse translation approach, if you will. That is, we start with clinical observations that can potentially be addressed in preclinical models. An example of that might be the observation that triptans, selective agonists for a particular serotonin receptor subtype or subtypes, are really only have efficacy in the treatment of migraine, despite the fact that these receptors are distributed throughout the brain, and so, throughout the body. And so one could certainly come up with hypotheses, as we have done, to explore the basis for that clinical observation and potentially learn something about mechanisms of migraine. Same sort of approach we've taken with the observations that my colleagues here in the regional anesthesia group noted in trying to monitor or manage opioid tolerant patients. That is, that these patients were not only tolerant to opioids, but that they were less responsive to local anesthetics. And again, that's something that we could potentially model preclinically. With respect to today's topic, the observations that sort of got us into this were the observations that compression injuries of the trigeminal nerve appear to be relatively responsive, at least for a limited period of time, to the anti-seizure medications, carbamazepine and oxcarbazepine. but compression injuries to somatic, that is below the neck injuries, are not. And examples of that would be, of course, classic trigeminal neuralgia, for which carbamazepine and oxcarbamazepine remain the primary therapeutic modalities, versus ulnar nerve entrapment or carpal tunnel syndrome, neither of which are responsive to carbamazepine. And so that was the clinical observation that got us going here. And that was brought to me by my colleague, was here at University of Pittsburgh Medical Center, is now up at Columbia. Ray Sakula is a neurosurgeon who treats trigeminal neuralgia patients. And most of the work that I'll be presenting today has been collected by Jorge Pineda Farias, a talented research associate in my group. So just going into this problem, one could look at trigeminal versus somatic nerves, and there's a number of reasons to suggest that the two may be different. And so from a therapeutic perspective, just the differences between those might account for the fact that there is this therapeutic selectivity of carbamazepine. There's some older data to suggest that the developmental origins of the trigeminal ganglia versus the dorsal root ganglia, which give rise to the sensory innervation of structures above the neck in the case of the trigeminal, or below the neck in the case of the dorsal root ganglia, may be different. The more recent seq analysis, RNA-seq analysis, suggests that might not be the case, but there's certainly differences in terms of somatotopy, with the DRG innervation having very different patterns than the trigeminal, and that there is at least some crude somatotopic organization within the trigeminal ganglia, and that really has not observed within the dorsal root ganglia. Certainly, there's differences in targets of innervation with unique structures in the face and head that really don't exist below the neck. The eyes, teeth, etc., would be differences in targets of innervation. The organization of the central terminals, trigeminal tends to be more rostrocaudal, whereas the somatic innervation is dorsal ventral in terms of the differential patterns. And most relevant is the observations. This is some recent results out of Rutgers suggesting that there's a differential response to injury. And they just did a gene expression analysis to show that with the same types of injury, you get different patterns of gene expression in dorsal root versus trigeminal ganglion neurons. So the first question we ask is, can we model this clinical observation preclinically? We use rats, both males and females. We use the chronic constriction injury, which is a loose ligation of either the infraorbital nerve or the sciatic nerve, to model a nerve compression. I'll be certainly circling back on that because I do not believe that the chronic constriction injury is a good model of trigeminal neuralgia per se, but that's what we started with. The methods, we had behavioral assessment with the oral facial pain assay and von Freiher's, compound action potential recording. I'll show you some of these data, and then quantification with PCR and Western blot. Rats were randomized to groups defined by nerve injury and drug treatments, and the experiment was blinded to treatment. For those of you that aren't familiar with the oral facial pain assay, there's actually two of these that are commercially available. I've got no financial relationships with either company, but it's Stolting and Ugo Basile. Actually, you can buy Ugo Basile products through Stolting. But basically, it's a box, so it's relatively objective. You can put the animal in it, walk away, and there's a port that the animal quickly learns to poke its nose through to get access to a sipper tube. We use 5% sucrose solution, and we don't have to even food deprive. There's enough of a reward to the sucrose that the rats will poke their nose through and access it. And you can occlude this opening with these tungsten wires that can increase in terms of density and therefore the mechanical force that's applied. And if you just monitor the cumulative contact time, so this is set up to objectively measure every time the tongue touches the sipper tube, closes the circuit, and so you can quantify the duration and number of contacts. You can, over a 10-minute period, show that there's a relatively consistent increase in terms of the number of contacts and no difference between males and females. And so we just plot total contact time across this 10-minute window. In naive animals, no real differences between males and females. Sham injury doesn't do anything. Both the chronic constriction of the infraorbital nerve with animals receiving either nothing or the vehicle for carbamazepine has no effect on the behavior. The advantage of this model is that you can see whether the drug itself has any influence on the behavior. And so the sham animals that have received carbamazepine, the drug doesn't influence the behavior. Naive animals, the drug doesn't influence it. But in the CCI animals, the 50 milligrams per kilogram completely reverses the hypersensitivity in males and almost completely reverses it in females, and almost completely reverses it in males. And the three-way analysis of variance would show a significant sex by injury by treatment interaction with post hoc analyses shown there. For the somatic tissue, and again, this is what makes this not such an ideal comparison, but still is a reasonable first approximation. We use the traditional von Freiherr test. Animals are put in these cages where we can get access to the ventral surface of the hind paw. And if we do a CCI injury, you get a dramatic decrease in the von Freiherr threshold, that is the mechanical force that the animals will withdraw from. And if you just plot the von Freiherr over time relative to drug treatment in sham versus animals that receive the drug produces, there certainly is an injury effect, that is, the threshold is down in both males and females. The drug has a slight increase in the area under the curve in both males and females, but there's no significant interaction, and the effects are relatively minor. So overall, we would argue that these results are consistent with the clinical observation, where carbamazepine has a therapeutic effect in the compression of a trigeminal nerve, but really minimal effect, if any, in a somatic nerve. So the next question we asked is what's the site of action for this? And we used compound action potential recording, hypothesizing that the changes in the peripheral nerve were responsible for the therapeutic actions of carbamazepine, which, as many of you may know, is a volscicated sodium channel blocker. And we can do a dose-dependent application of carbamazepine. This would be an example of a compound action potential where we can look at the A wave. We didn't really quantify the A delta, but the C wave would be the slower wave following along. And with increasing concentrations of carbamazepine, you can block the A wave and the C wave. And if you just plot fractional block versus carbamazepine concentration, you get a significant leftward shift in the concentration response curve in the injured nerve, suggesting that there's the emergence of an increase in potency with the injury. And this is true of the ION, but not the sciatic nerve. So in the same sort of recording paradigm with animals with the sciatic nerve, there's no impact of the injury on the potency or efficacy of carbamazepine. And these data are just plotted in a slightly different way, with carbamazepine concentration, with the males versus females, sciatic nerves versus infraorbitals, sham versus CCI. And again, you can see this dramatic, it's almost an order of magnitude drop in or decrease in potency, increase in potency for both the A wave and the C wave. Within the C wave, a sex by nerve by injury interaction between the two, or between the three. So the question then is, what's the basis for this differential therapeutic effect of carbamazepine? As I suggested, the data in the literature would suggest it's a use-dependent voltage-gated sodium channel blocker. There is at least some evidence of a differential sensitivity among voltage-gated sodium channels to carbamazepine, and therefore we hypothesize that the differential distribution of voltage-gated sodium channels following injury is what conferred this shift in potency and therefore the therapeutic efficacy of the drug. And so that's what we asked: is there a nerve injury change in voltage-gated sodium channels? This is a very complex slide, but hopefully just visually you can see what's plotted here are data from sham and CCI injured ION, as well as that's infraorbital nerve versus sciatic nerves. And you can hopefully see that there's not only an influence of injury, as you can see, say a drop with this ICA compound in both A wave and the C wave, or and a difference between the sciatic nerve versus the infraorbital nerve, supporting the notion that there really are differences between, even at baseline, between the trigeminal versus somatic nerves in terms of the voltage-gated sodium channel that are functional, enabling propagation along the nerve, but that injury has a differential influence as well. And this is just the targets for these different drugs, and the fact that I'm not including, we've got a 1.3 blocker in here that had no effect, but because the 1.3 blocker didn't have an effect in the nerve, but the 1.1 that the ICA did, we concluded that 1.1 might be the emergent target, if you will, for following nerve injury of the trigeminal, but not the somatic nerve. And this is just the max fractional block with this ICA against the A wave and the C wave. And again, there's a nerve by injury interaction with the emergence of this, the increased fractional block in the ION, but not the sciatic with nerve injury. And we can see, at least with this anhydro TTX, an influence on the A wave, but not the C wave, arguing again that not only are the nerves different, that is somatic versus trigeminal, but A waves and C waves may also be different in terms of the underlying channels enabling propagation. So to confirm that NAV, or at least to further pursue the notion that the NAV 1.1 might be involved, we did Western blot of the nerves, sciatic versus ION, and there appeared to be a significant and selective upregulation of 1.1 in the infraorbital nerve that we did not see in the sciatic nerve. So no change in the infraorbital nerve. In contrast, in the sciatic nerve, there was a significant increase in 1.3, again highlighting the differences between trigeminal and somatic nerves. And then we can ask, functionally, does the 1.1 contribute to the hypersensitivity? And we did that by using a local injection along the nerve of the ICA, just to block the infraorbital nerve, and ask, does that reverse the mechanical hypersensitivity in this in the oral facial pain assay? Again, with the CCI, you get this decrease in contact time. Sham vehicle doesn't do anything. Sham ICA, again, there's no effect of ICA alone, but the ICA reverses the hypersensitivity in the CCI animals. And we can do a comparable experiment just to confirm that there's really no increase in potency for ICA in the sciatic nerve injury. These are all showing the hypersensitivity that's really has no influence when we apply ICA. And I'm not showing the controls here, but we've got controls with TTX or other blockers to show that we can reverse the hypersensitivity. So the summary so far is that there's an injury-specific difference in the efficacy of carbamazepine. There seems to be a sex difference in terms of the magnitude of the effect. There's a nerve-specific difference in terms of the potency of carbamazepine. Again, a sex difference in terms of the magnitude of the effect. And these changes appear to be associated with an increase NaV1.1. There's an increase in protein in the nerve. This is an interesting question that we're still pursuing. But what it looks like to us is that we have local translation in the nerve as opposed to a change in transcription and the trafficking of the nerve within the axons. And there's a shift in the therapeutic efficacy. In this case, there was no difference in terms of males versus females in terms of the overall therapeutic efficacy. So in terms of next steps, as I suggested or alluded to at the start of the talk, we're really pursuing a more relevant model of trigeminal neuralgia. For those of you not familiar with the disorder, the International Classification of Headache Diseases or Headache Disorders is pretty clear on how trigeminal neuralgia is defined as either classic, secondary, or idiopathic. And what really separates trigeminal neuralgia from other types of neuropathic pain syndrome is this paroxysmal pain that's triggered. And that's really what the patients are most disabled by, is this just incredibly intense pain that can last from a couple of seconds to a little bit longer, but it's just so intense that basically you're not thinking about anything else while this is going on. There can be some concomitant continuous pain, but it's the paroxysmal pain, and paroxysmal pain that's triggered. Most of these patients have sites on their face, generally in the V2 dermatome, that makes them hyperresponsive to stimuli. And so the problems with the current models is that there really is no evidence of paroxysmal attacks. That is, that if you probe with von Freiher's, for example, or in the assay that we showed, the animals are basically hypersensitive all the time and don't show this, what we would predict would be consistent with paroxysmal attack. And they certainly have no evidence, at least again with the models that are available, of the sort of trigger points that you see in the patient population. The current models that are out there, we use this chronic constriction injury of the infraorbital nerve. More recently was this foramen elasticum compression model, where the central portion of the ganglia is accessed via this foramen at the base of the skull to enable a selective compression. And what we've been working on developing is this trigeminal root entry zone compression, which we would argue is really what may be the critical feature that distinguishes trigeminal neuralgia from other types of neuropathic pain syndrome. And this is just a MRI of a patient where you can see the type of compression that occurs with actual deformation or real constriction of the trigeminal nerve as it's transitioning into the brainstem here. This would be the ipsilateral side where you can see the nerve entering unimpeded. And the current strategy for these patients is either an ablation or what is referred to as a vascular decompression. What's unique about this site is that there's a transition between what would be centrally myelinated, that is with oligodendrocytes, and peripherally myelinated, which would be by Schwann cells, innervation of the nerve right about at that transition zone. So you can see that both in the rat and the human. The brainstem is here in the human. This is the peripheral nerve. Here's the ganglia. But you can see that there's a relatively long extension of the oligodendrocyte myelinated component. This is just two different myelin stains, one for central versus peripheral. And you can see the same thing in the rat, again, with the trigeminal ganglia. And there's some data from an older study to suggest that there's a correlation between the prevalence of these sort of compression-induced abnormalities that are associated with these sort of paroxysmal attacks, with the trigeminal nerve having the most common manifestation of these types of pain syndromes, where this would be the total length of the central process of the nerve, where the centrally myelinated portion is the longest for the fifth cranial nerve, which innervates the trigeminal ganglia, and of course provides the sensory and nociceptive innervation of the face and head. Facial nerve would be the next, which would be the next longest, whereas these other nerves really have a very relatively small centrally myelinated portion. And so again, there's at least correlative evidence to suggest that it's this length and therefore possibly the site of the compression is relevant. And so we've been working out strategies to access this central portion of the nerve as a way of getting at the forming the compression. The next challenge that we've been facing then is really how to assess the presence of paroxysms, and the two approaches that we're pursuing at the moment are looking at autonomic fluctuations and the Grimace scale. I'll say a couple more words about the Grimace scale in a second. But in terms of autonomic fluctuations, these are data collected with a galvanic skin response from trigeminal neuralgia patients, where we're either using an electrical shock to drive a nociceptive response, or triggering it by lightly touching their trigger spot. And you can see very dramatic differences, or at least increases in the galvanic skin response in response to the triggered attacks, suggesting that we could possibly use these autonomic fluctuations in the preclinical model to monitor the presence of a triggered attack. In terms of the Grimace scale, Jeff Mogul had developed this. People now refer to it as the Mogul Grimace scale, but he described it as the mouse Grimace scale. And originally, this was scored via individuals. That is, you would videotape, and you'd have people, and he used undergraduates, basically crunch through 10-minute blocks of video, frame by frame, scoring when the animal was facing you, positions of the ears, the eye position, the nose position, et cetera, and you can generate a score based on the position of those things. That's been automated now by now why am I blocking it? Oh, Mark Zilka and his colleagues at UNC Chapel Hill. So there's basically an AI mediated or driven recognition software that can then automatically score this. And these are data that were collected by a talented graduate student in our group, and Nicole Sheff, the PI that he's working with at the moment, on using the Grimace scale, and just frame by frame, where the score is plotted in or is coded by color. So the higher the score, the more Grimace, in theory, the more pain behavior is supposed to be reflected. This is an oral cancer model that they used, but you can see that the behavior is not consistent over time, but there's really relatively low or no Grimace during the baseline. Sham animals 15 days later show no change, but you can see a dramatic increase in grimacing in the animals that have this cancer injected into their tongue. And so we've been piloting this approach to be able to quantify the emergence of bouts of spontaneous activity in the animal. And so just to wrap up here, to thank the folks that did the work. Again, here's Jorge, who collected the data. This is the rest of the group working on different aspects of the study. Lucky enough to have a number of talented people in the lab. I had Grunenthal on here, but that's relatively, I think it's outside of the 2 years window. So, and none of the data I'm showing today were supported by that. But thank you all for your attention.
All right, thank you, Michael. That was really interesting presentation. Yeah, we've had some great questions. Just a reminder to everyone else in the audience to basically for Professor Gold's presentation, if you've got any questions, please enter your questions into the pane, and then we'll collate it all at the end. Okay, so Now I want to turn to the next presentation, so I'd like to welcome Dr. Tony Rush. So he's Director of Neurosciences at Metrion. So Tony's worked in the ion channel field for over 25 years across both academia and industry. And he received his PhD from University of Dundee, was a postdoc at Steve Waxman Lab in Yale, and then he worked across a range of biotech and pharma, predominantly focusing on ion channels and pain. He was a founder at Metrion, and his responsibilities including leading the collaborative drug discovery projects and also managing neuroscience activities at the company. So over to you, Tony.
Thanks, Eddie. So the first part of my talk is to be concentrating on particular sodium channel assays that we can perform at Metrion, and then particularly focusing on those related to those in sensory neurons. So this first slide just highlights that Metrion has the full cell line panel of human sodium channels available, so ranging from NAV 1.1 right up to NAV 1.9. And these assays are validated on both manual and automated patch clamp platforms. There's increased interest recently subsequent to the NAV 1.8 success of suzetrigine from Vertex after their approval for treatment of moderate to severe acute pain in adults. So if I just highlight a couple of the automated patch assays in this slide. So in A, we have human NAV 1.8 single hole currents recorded on QPatch. We have the current recorded on the left-hand side, or an example, so holding at -100 and then depolarizing to 0 millivolts to activate the current. And then on the right-hand panel here, we have exposure to a vehicle of 0.3% DMSO over time during these regular depolarizations. And then we have P1 and P2. So P1 is the resting state, and P2 is to assess the inactivated state. So showing really, really good stability over many minutes on the QPatch single hole assay for NAV 1.8. And on the right-hand side, we have our relatively new NAV 1.9 cell line, and this is data recorded in multi-hole, and this is on the Cube platform, so our 384 platform, as opposed to the QPatch, which is 48 wells. So on the left-hand panel, we're showing well-clamped sodium currents from the cell line in multi-hole mode, and then you can see the voltage dependence of activation on the right-hand side, so showing it peaking at a fairly hyperpolarized voltage, which is what you would expect for NAV 1.9. So showing the correct biophysics of the channel. But just to remind everyone of the interplay of sodium channels in sensory neurons. So we have a variety of different channels expressed in sensory neurons. And in A, what I'm highlighting is the contrasting voltage dependence of TTXR and TTXS currents. So the TTXR we're referring to here is likely to be carried by NAV 1.8. What I'm highlighting here is that the TTXS currents tend to be have fairly hyperpolarized voltage dependence of activation, and also fairly hyperpolarized voltage dependence of steady state inactivation as well. Whereas the TTXR current, likely to be carried by NAV 1.8, has much more depolarized voltage dependence of both activation and steady state inactivation. So this obviously affects the way that the channels behave when they're supporting firing of the neurons. But also to point out in panel B, so this is data from the Waxman lab, so showing repetitive firing role for NAV 1.8. So in the left-hand panel, we have an example wild-type neuron where the cell is being activated for maybe a second to activate a train of action potentials in the wild-type. But when you do a similar train or a similar input for the NAV 1.8 knockout, you see that the train is no longer present. So it appears that the repetitive firing in many DRG, or small DRG neurons in particular, is related to the presence of NAV 1.8, as it's not present in the knockout. And then in C, what I'm highlighting is the potential roles of NAV 1.7 and 1.8. So this is from a recent review from, again, from the Waxman lab. So the NAV 1.7 is thought to be potentially the channel that basically can bring the or amplify inputs into the neuron and brings the neuron to threshold, which then activates other sodium channels, which are then responsible for the upstroke. So this is partly due to the different voltage dependence of NAV 1.7 being more hyperpolarized than NAV 1.8, but also potentially the fact that it responds greater to ramp currents. So these slow depolarizations is more likely to activate NAV 1.7 than 1.8, and may be involved in this getting to threshold. But in D, more recent data from the Angelica Lampart lab at the University of Aachen shows a potential greater role for NAV 1.9. So this is modeling studies that the Lampart group has done. So it shows potentially that NAV 1.9 could actually be involved in both the upstroke and also the repolarization phase, the hump of the action potential. So it's possible in these modeling studies suggest that NAV 1.9 may have an additional role in the actual action potential itself, whereas previously it was thought to be only involved in potentially setting the resting membrane potential. So to run through some of our assays, particularly in DRG sensory neurons. So we're very sort of conscious of setting up robust assays. So what we're showing here on the left-hand side in A is a family of NAV 1.8 currents. So we make sure that we're always checking that we have very, very good voltage control of the currents. So you can see the family in A, and then the current voltage relationship is shown in B. So very little leak, and then the peak of the current is, as I previously suggested, is relatively depolarized, peaking at maybe 0 or plus 10 millivolts. And then in C, we've set up looking at current stability over the time. So we're evoking a peak current perhaps every 10 seconds, and then you can then put together an IT plot over time and record for maybe up to 15 minutes, showing really nice stable recordings for this current. This then allows us to really well test pharmacology. So pharmacological investigations here. So in A, what we're doing is again an IT plot showing peak current. So we make sure we get a nice stable baseline, and then we're putting increasing concentrations of compound which reduce that current. The associated currents are shown in B, color-coded to match the IT plot. And then you can put together, in this case, a composite IC50 curve from multiple cells if you want to have multiple concentrations. Moving on to NAV 1.7. So this is, instead of using voltage clamp, this is now using current clamp recordings to investigate NAV 1.7 and its pharmacology. So here what we're doing is a relatively short ramp response to evoke a train of action potentials. So these are two different example recordings. So you get a certain number of action potentials, multiple action potential firing and control, which in the presence of an NAV 1.7 inhibitor, in the top example, this pretty much eradicates that train of action potentials, which then nicely washes off afterwards, and we get a nice return to similar recordings to what was shown in control. But in example 2, we have a nice train of action potentials induced by the ramp, but then the train of action potentials is delayed to later in the ramp, but you still get that train of action potentials. So that those multiple firing is probably related to the presence of NAV 1.8. The 1.7 inhibitor, it's suggestive that it is decreasing that, increasing that threshold to firing. And again, we have nice wash off on the right hand side. But in the presence of TTX, we can also record both NAV 1.8 and 1.9 together. And what we're showing in this slide is subtracting out the NAV 1.8. So this top panel here is showing holding potential at -120, depolarizing to a range of potentials to activate the currents. This is the total TTX resistant current in the cell. We're then moving to a holding potential of -70, and then repeating the family of currents. This largely activates the NAV 1.8 current, and then we can subtract these out, and this is shown on the right hand side. So we now have the cell line of NAV 1.9, and we can also follow this up with recordings in DRG neurons. Moving away from sodium channels, I'd like to give an example of functional effects that to do with KVs 7.2, 7.3, another popular pain target, which is still being investigated at the moment. So these recordings are not from DRG, they're from trigeminal sensory neurons. And what we're showing in A is resting membrane potential recordings. And we're using retigabine, a potentiator of this current. So what should happen as you potentiate this current, you should get hyperpolarization of the RMP, which is exactly what happens, and then returns to baseline. This, of course, doesn't occur in every single cell because you don't always have expression of KV 7.23. So we get a range of activity, whether depending on whether the channel is expressed or not. And this right-hand panel here shows the change in resting membrane potential. So you can see the range of activity across the different neurons that were recorded. We can also examine the effect of this kind of compound on multiple firing using retigabine. We have a train of action potentials from trigeminal cells in this top panel, which is then inhibited by 10 micromolar retigabine, but leaving that first action potential untouched, which is what you would expect for a potentiator of this current. But this happens in every cell that was recorded that has multiple firing in the trigeminals. And then moving on to calcium imaging and potentially combining this with manual patch clamp and identifying different targets in sensory neurons. So here what we've done is we've used the patch scope system, which is a combined calcium imaging and manual patch platform. And then we've assessed the expression of TRPA1, TRPV1, and TRPM8 in our population of DRG neurons. What you would expect from the literature is that around 25% of cells express TRPA1, and we get around 27. TRPV1 should be around 70%, which is also replicated. And TRPM8 on the right-hand side is much rarer and is below 20% expression. One reason we were looking into this was that this raises, when you're trying to do experiments on targets such as TRPM8, this may be very difficult using just manual patch on its own, but we can potentially identify neurons using this method using imaging. So this left-hand panel here shows that a neuron is responding to menthol to identify the expression. We can then go and patch that neuron in either in B, current clamp mode, or voltage clamp mode in C, and show and then record a response to menthol from that neuron. So this means that we're no longer doing a fishing expedition looking for expressors, and we can then basically identify those expressing neurons and then go and patch those cells. Just my last slide is showing that Metrion also has, aside from sensory neurons, has a whole range of different neuroscience assay services, ranging from using a wide variety of cell lines that we can investigate with a variety of different platforms. We also have central neuron assays from rodents, and then also human iPSC-derived neurons. And just some example data from iPSC-derived glutamatergic neurons is shown on the right-hand side, showing ligand-gated NMDA currents and the associated EC50 curve from those results. So thank you very much for listening.
Right, thanks, Tony. That's great. So, well, we can now sort of move over to questions for the presenters. So, yeah, so keep posting questions in the question pane, and we'll answer as many questions as we can. Okay, so yeah, we've got our first question. Okay, is really about from you, Michael, is about the sex differences. So firstly, yeah, just further insight into why why you think there is such a difference between male and female in terms of carbamazepine response to the facial nerve injury, and how does this translate? Is that seen in the clinic as well? Are there clinical differences with carbamazepine?
Yeah, so that's a good question. The why is always a tough one. There certainly is a sex difference in terms of the manifestation of trigeminal neuralgia that's been described clinically. This pain disorder is one that occurs relatively late in life, and in contrast to many other pain syndromes like migraine, temporomandibular disorder, painful bladder disorder that occur sort of increase in frequency and prevalence post-menarche and resolve with menopause. Trigeminal neuralgia increases in menopause, and again, or sorry, after menopause, and again is more common in women than in men, arguing that at least the sort of dynamic or activational role of gonadal hormones may not be a real driving factor there. We're using relatively young animals, so we're not using an age that would be necessarily comparable to the neuropathic pain patients. It's an interesting observation that the way that the drugs are used in the patients, detecting a difference in potency would be potentially difficult in terms of just the titration rate, and whether it'd be able to differentiate. That my colleague Ray Sakula wasn't aware of any data and really no major observations in the populations that he's seen of a difference that way. So it may be a species difference, but it certainly is something that we're pursuing in terms of potential mechanisms that may suggest the need to sort of differentially treat men and women suffering from this disorder. Long-winded answer to say I don't really know.
Okay, brilliant. And then another question was about somatic pain. So have you looked, do those differences between facial and somatic pain disappear when you look at other spontaneous pain models like burrowing for somatic pain?
Yeah, so we've been using more and more of these, functional assays, burrowing, home cage wheel running, nest building, and those certainly support the oral facial pain assay data, and you can see deficits in the neuropathic pain animals that are reversed with the drug treatment. So I'm certainly in favor of using those additional endpoints, particularly if they can be done in the home cage. The nice thing about the oral facial pain assay is it's quick, and so we don't have to monitor all night. The burrowing assay is a nice one because it's relatively easy to quantify. It just takes a little more time clean up the cage after the fact. But yeah, we do see we have been rolling those in more and more frequently, and we do see basically the same pattern of results that we see with these. In the case of the oral facial pain assay, would be an operant assay.
Okay, brilliant. So we've had quite a few questions about the role of NAV 1.1. So have you looked at expression in different subtypes? You know, because 1.1, 1.6 tend to underlie A fiber, action potentials rather than C fiber. So have you looked whether it's upregulation in a particular subtype?
Yeah, so we've basically been, I just showed you a relatively narrow panel, but we screened all 9. And the differences that really jumped out were the ones that I showed, where in the trigeminal nerve and the infraorbital nerve was this increase in 1.1. That slide that I showed did highlight the presence of, or at least pharmacological sensitivity to 1.1, and we can pick up bands even in the absence of nerve injury. So it isn't that it's going as was originally described by Steve Waxman's group for NAV 1.3, where you really have no expression in the adult. You injure the nerve, and then you get this upregulation of 1.3. That doesn't appear to be the case for 1.1. But its relative contribution to action potential propagation changes. And like I say, we screened both pharmacologically and looking at mRNA and protein for all 9 of them. And there were subtle changes in some, but the most dramatic changes were in the somatic nerve, 1.1 was upregulated. Sorry, 1.3 was upregulated in the trigeminal 1.3. Sorry, 1.1. Apologize for misspeaking. That isn't to say that these other channels don't contribute, but at least our results in the infraorbital nerve would suggest that the sort of canonical, 1.6 mediates the action potential propagation in A fibers, and maybe some other channel subtypes mediate it in the C fibers. Didn't, at least in our hands with the pharmacological tools that we used, 1.6 contributed, but wasn't, that 90% of the action potential propagation was dependent on 1.6. It appeared to be a combination.
Okay. And then what's your thoughts about why does NAT regulation of 1.1 lead to greater sensitivity to carbamazepine? What's the thoughts?
Yeah, back with the why questions. Yeah, so I could certainly wave my hand around the why. I mean, the assumption that as Tony really nicely showed in terms of the biophysical properties of these channels, is that they're different. They're different pharmacologically, they're different biophysically, and my sense of what is happening in the axon to enable propagation is really a balance between the needs of the organism to enable propagation at a given frequency and fidelity. And so you certainly could envision a situation whereby, and we had hypothesized this, way back in the early 2000s when we described a nerve injury-induced upregulation of NAV 1.8 along the axons, that there was a sustained depolarization as Wallerian degeneration was taking place and immune cells were had moved in to help clean up the injured nerve, that the axon might be depolarized because of that, and therefore you needed a sodium channel like NAV 1.8 that could withstand a relatively large depolarization and still enable action potential propagation. And so in an axon where 1.8 isn't normally functioning very, very significantly or contributing very significantly to the action potential propagation, the context of what might be a local inflammation induced depolarization, you might need a little more 1.8. Whereas you can certainly envision other situations where if the membrane were hyperpolarized, where if the pattern of firing was changed and influenced the signaling. Carol Torsney in the UK has been doing some really interesting work on action potential slowing, and certainly these channels and how they contribute and the impact of action potential slowing on the integration of information at the central synapse. The subtle biophysical property differences between these channels can play a significant role in changing that response.
Okay, great. Thanks, Mike. Right, so we've only got time for one more question, and actually I think I'll ask Tony this one, which is, I think, is quite a tough question. So yeah, in terms of 1.9, what should Angelica Lampart's data suggesting 1.9 has a role in the upstroke? What about repetitive firing? Do you think 1.9 has a role in repetitive firing?
Well, I think the persistent part of the NAV 1.9 current is likely to undergo substantial inactivation, but perhaps the transient current at more depolarized voltages could well still be involved. The transient part of the current hasn't actually been investigated in depth, hasn't been well characterized, and of course the best way of investigating this is to have a selective inhibitor, but that currently doesn't exist. So we can hope that the future brings more selective ligands, and then we can start investigating some of these things in more detail.
Okay, fantastic. Well, okay, great. So well, thank you for everyone sort of posting the questions. That's great, and thank you to Professor Gold and Dr. Rush for their great presentations, and to everyone who's joined the webinar. So our next webinar on the 30th of April will present a case study about NAV 1.9. So it's very timely to ask that question as a drug target for the treatment of pain, and I'll be presenting alongside David Bolmer from University of Cambridge. And you can register on our website events page. So thank you again for joining us, and yeah, the webinar will now close. Thank you, everyone.